Okay, so this is actually our last lesson on this topic. So we've actually had kind of three subtopics within a bigger organic chemistry topic. So this is our not only our last lesson about polymers, it's also our last lesson of the bigger organic chemistry topic. And that big organic chemistry topic started with the stuff we learned about crude oils and fuels. So everything we've learned since our first lesson about crude oil and fuels through to today's lesson is part of a big organic chemistry topic. My plan is that in tomorrow's lesson you will do some consolidation, recap, revision of this big topic. It is quite a big one. We've done a couple of sort of consolidation lessons, live consolidation lessons along the way as well to try and help with that because it is a big topic. And then next Tuesday, let me just double check. Yeah, next Tuesday, that's the 14th of July, I'd like you to complete the test on this topic. If you could try your very best to get that completed on Tuesday and submitted so that we can review that before the end of term, because we will have just one lesson left on the Friday of that week. Then that finishes off this topic so we don't forget it over the summer. Obviously, in the end, we're going to have to remember everything for the end of year 11, but otherwise we'd be starting the new year with a test, which is not ideal. So right now, let's get this lesson started. You should have the title Natural Polymers in your book. If you haven't got that in your book, write that in there now, underline it, and have a go at the three starter questions that are on this slide. So pause this, answer those questions, press play when you're ready to go through the answers. So first question, what is the difference between an addition polymer and a condensation polymer? Well, the difference is when you make an addition polymer, all you are making is the polymer. The polymer is the only product. Condensation polymers, you make that long chain polymer, but you also make another small molecule as well, usually water, but it's another small molecule as well as the polymer you're forming. The structure of ethanoic acid. Let's have a go and see if I can draw this on the screen. So ethanoic acid, F means we have got two carbon atoms. So we need two carbon atoms there. So that's this part of the name tells me we've got two carbon atoms. The oic acid tells me it's a carboxylic acid. So the carboxylic acid functional group has a double bond going from the carbon to an oxygen, and then a further single bond going to an OH group. If we're doing a full displayed formula, we should also have the bond between the oxygen and the hydrogen. I'm just doing a sort of a semi-shortened version there. And then this carbon here on the right hand side, that's okay because we've got four bonds coming from that one already, but our other carbon here, we need to add on our hydrogen such that we've got four bonds coming from that carbon all together. So one on the top there, one on this side, and another one underneath as well. And there's our ethanoic acid. Let's just get rid of that arrow because we don't need that there. There we go, let's get rid of that. Okay, and finally it says, what does decreasing particle size, so why does decreasing particle size increase the rate of reaction? Well, if you have smaller particles, this is if you keep, I'm assuming you'd have the same mass, but if you have the same mass of reacting solid in smaller particles, you are increasing your surface area. So the first thing is you're increasing surface area, which means you've got more surface exposed for collisions. So you're going to have a increased frequency. Remember that word, increased frequency of collisions or more frequent collisions, and that will give you a increasing rate of reaction. So higher surface area, more frequent collisions will give you a higher rate of reaction. Let us move on. So here are the things that we should know by the end of this lesson today. First of all, you should know that sugars can undergo polymerization in living things to make polymers such as starch and cellulose. Secondly, you should know that proteins are polymers made of amino acids. And last of all, you should be able to explain how nucleotides are arranged in DNA. Now, some of this, I'm not quite sure how much this is covered on the biology specification, but this is kind of an area where biology meets chemistry. It's kind of biochemistry. So some of this you may touch on in biology as well. So the first thing I want to look at is the natural polymers that are made from glucose. So I'm going to say just now is you may want to pause this video as you go through and make some notes. You might just want to listen to the video all the way through and then go back through the slides to make some notes, or I have put the page numbers from the textbook onto Classroom as well, so if you want to just listen to the video and then use the textbook to make some notes, I'm going to leave it up to you what notes you need to make on this, but the important words I've highlighted here in red, but obviously by themselves they wouldn't make much sense. So I'm just going to carry on today and I'm going to let you decide what notes to make. So you already know that plants make glucose during photosynthesis. You certainly would have covered that in biology. And glucose is a monosaccharide, which means that it's made of one sugar unit. I've shown over this side over here, I've just shown one unit of glucose, so that's a glucose molecule over there. Monosaccharides, however, can join together in long chains, so they can form polymers basically, and the polymers that they form we call polysaccharides, so they form these polysaccharides, and it's an example of a condensation polymerization reaction. So they undergo condensation polymerization, join together, and the monosaccharides become polysaccharides. And it's even, the name is kind of like in a similar way to the way that we have named our polymers before. We just put the word poly in front, but we just haven't bothered with the brackets this time. Now, this is slightly confusing because we've got two things that are examples of polysaccharides. So cellulose and starch are both examples of polysaccharides. So they are both formed from glucose units joining together to make polymers. So let's just read what it says here. They're both formed from glucose molecules joining in condensation polymerization reactions. In cellulose, the glucose units are joined in straight lines, or in straight chains. In starch, the glucose units form more branched chains, and I've got a little bit of a sort of pictorial representation there at the bottom. Okay, so sugars, they undergo polymerization and they are glucose in particular, and they can make starch and cellulose. So already we've covered the first thing on this slide. Let's move on. So amino acids and proteins, things you'll definitely hear about in biology. Proteins are other natural polymers, and the monomers of proteins are amino acids. So they've got two functional groups, these amino acids. They've got a carboxylic acid functional group. So over here, I've got my carboxylic acid, or my carboxyl group, with a C double bond O and the OH, so that carboxylic acid group or the carboxyl group. On the other end is an NH2 group, and that's called an amino group or an amine group. So that amine group is on the other end of our amino acid. Incidentally, this letter R here can be a number of different things. In the very simplest amino acid, it's just a hydrogen, but often it's a carbon joined to other things as well. So we can have various different things where that letter R is, but there's always an H there at the top there. So that's always an H, the R changes. Now, the amine group acts as a base, and it can react with the carboxylic acid group from another amino group. And if you think back to our chemical reactions topic, we know that bases and acids can react together, and that's exactly what's happening in this reaction. Acid plus base gives us a salt plus water, and water is lost in this reaction. So it's another example actually of condensation polymerization, because we produce water as well, and because we've got the two functional groups on opposite ends of the molecule, one functional group from one molecule can react with the other functional group on the other end of a second molecule, and they can all line up and form these long chains. And hopefully you can see here, so we've got two amino acids at the top here. We've got our carboxylic acid group there from one, we've got our amino acid group from that same one there. Let's just pick a second colour there. So we've got our amine group from that amino acid there, and the carboxylic acid group there. And of the ones in the centre, so we've got, let's just do a pen for a minute. So those two groups can react together because they've come close to each other. So the acid group reacts with the amine group. And when that happens, we lose water. So we're going to lose the OH from the acid, the carboxyl group, the H from the amine group, and form water. And then this carbon will become directly bonded to the nitrogen. So that carbon there will become directly bonded to the nitrogen, and that's what's shown with the structure underneath there. So you'll get this is just a dipeptide because it is two amino acids joined together. We call that a dipeptide. It's got this peptide bond in the middle, so that bit in the middle there, that's our peptide bond, or the actual bond there in the middle of that is our peptide bonds, and that's the sort of group that joins it all together. So I've now scribbled on that and made it a little bit untidy. In case you are using this to copy things down, maybe we'll just get rid of some of those annotations just so it's easy to see, but I just wanted to explain how that all happens. Fabulous. Okay, I mentioned this term dipeptide before. So if you've got two amino acids just joining together, then you get a dipeptide. If you've got three amino acids joining together, we call it a tripeptide, and so on. When you get lots of amino acids joined together, we call it a polypeptide. So all a polypeptide is it's lots of amino acids joined together in a long chain. And the link there to proteins, because this is where it can be a bit confusing. What's a polypeptide and what's a protein? Well, this bit at the top here, you see the peptide or the polypeptide is the long chain. They tend to fold in specific 3D structures, and it's the protein is this sort of 3D shape formed by the polypeptides. So at the bottom there I've said the protein is one or more polypeptide folded into a 3D shape, and different proteins in the body or within living things have very specific shapes. Okay, so we've had glucose forming starch and cellulose, so there's starch and the cellulose, and then we've had amino acids joining together to make proteins. So in each case we've got our monomer, which was glucose in the first example, making starch and cellulose, and in the second case here amino acids were our monomers, and they made our polypeptides, or our proteins were our polymers, if you like. Last one we need to cover is how nucleotides are arranged in DNA. So DNA is short for deoxyribonucleic acid, and it's a large molecule which is essential for life. You will have heard of it before. DNA encodes the genetic instructions for the development and functioning of living organisms. So all living organisms have DNA, and even some viruses have some DNA as well. DNA molecules are two polymer chains made from 4 different monomers called nucleotides. So this time our monomers are called nucleotides. And they join together in these chains and they form this double helix shape that we are very familiar with for the shape of DNA. Okay, so there's just a little picture of some DNA just there. Now, if we look closer in at our nucleotides that are our monomers for the DNA, each nucleotide has a part which is a sugar, so we've got our sugar there, and then it's bonded to a phosphate group over here, and it's this bit, so this phosphate group sugar bit, that forms like the backbone of the chains. But attached to the sugar, we've also got these bases, and there are 4 different bases in DNA. So one of these 4 bases will always be attached to the sugar. It could be adenine, which we give the capital letter A, guanine, which is given a capital G, cytosine, a capital C, and thymine, a capital T. But it's this, as I said, it's the part there with the phosphate and the sugar that forms the backbone of the DNA helix, if you like. So they join together rather like this. So the DNA is basically a polymer of sugar, phosphate, sugar, phosphate. So we get this alternating phosphate, sugar, phosphate, sugar, phosphate, sugar. And each sugar has got a different base attached to it, or can have any one of those 4 bases attached to it, basically. Okay, this diagram is just trying to show you a little bit more of how the structure lines up. Now it's flattened out the 2 helixes that intertwine this alpha helix, this double helix that is so characteristic of DNA. And the reason it's held in this double helix is because there's something called hydrogen bonds between pairs of the bases which are attached to the sugar. And specific bases will form hydrogen bonds with specific other bases. A and T will always form a pair, and C and G will always form a pair. You'll never get a T hydrogen bonding with G or an A with C. So it's always A and T, C and G. And the way I remember that, this is more biology, you don't really need to know this for chemistry, is it's the ones with the straight line letters go together, and the curvy line letters go together. It's just the way I remember it. So hydrogen bonds, all that they are is they are a type of intermolecular force, and we've talked about intermolecular forces when we talked about bonding at the beginning or towards the start of year 10. If you want to pause and have a little look at this poster, so this is a poster that's freely available on the internet, and this tells you a bit more about the chemical structure of DNA. Got quite a bit of information on there. Feel free to pause this and have a little bit more of a look. It tells you the structure of the different bases. You don't need any of this detail for GCSE chemistry. It's here for interest and nothing else. And there's a nice little model of DNA showing that double helix structure and the rotating there. So hopefully you can see sort of in the middle there, we've got those sort of base pairs coming together, sticking out from the different sugar groups to form that structure. There we go. Fantastic, and that is it. So that's the natural polymers that you need to know about. If nothing else, you need to be very clear which monomers make which polymers. So I've just put that into a summary table. I would say that you do need a little bit more detail of that. As I said, you feel free to use these slides or this video to go back and make some notes if you haven't already paused it and done so. Alternatively, use the pages in the Caboodle textbook to make some notes for this lesson. Otherwise, that's it. So thank you very much for listening.